Bienvenidos sean todos a un nuevo webinar de la Facultad de Medicina, Mutaciones SARS-CoV-2, Implicaciones Epidemiológicas y Clínicas, impartidas por el profesor Nassiri. En nombre del personal académico, administrativo, manual y cuerpo directivo de nuestra dependencia, agradecemos su participación en cada uno de nuestros eventos. Aprovecho esta oportunidad también para invitar a quienes siguen esta transmisión en vivo a través de nuestras redes sociales para que nos dejen sus comentarios, preguntas y saludos en el apartado de comentarios de las publicaciones en vivo, mismos que serán comentadas al finalizar por el doctor Pablo González. El día de hoy contaremos con la presencia de un invitado de honor, el profesor Reza Nassiri, y nos hablará sobre las mutaciones SARS-CoV-2 desde una perspectiva de las implicaciones epidemiológicas y clínicas. El profesor es actualmente profesor de farmacología y toxicología en la Universidad de Michigan State. Sus intereses de investigación se centran en los campos de quimioterapia, de medicina tropical y enfermedades infecciosas, VIH-Sida, resistencia a los antibióticos y salud global. Pero antes de iniciar y cederle la palabra, quisiera ceder la palabra a nuestro secretario académico, el doctor Edgar García, quien nos dará unas breves palabras de bienvenida en nombre del doctor Carlos Castro, director de nuestra facultad.
Gracias, Juan. Welcome, Dr. Nassiri, Reza.
Thank you. Good to see you.
Thank you so much. It's an honor to share your experience about COVID, SARS-CoV-2. It's very pertinent because we are in the third wave to COVID, about the mutations to the Delta forms, no? So this is your house, and this is school medicine, and the name to the Dr. Castro, to Universidad Autónoma de Yucatán. For us is very important you talk about COVID.
Gracias.
Thank you so much. So this is your plataforma. Thank you. Gracias, doctor Pablo. Podemos comenzar. Gracias.
May I start? Can you hear me?
Yes, we can.
Okay. Well, thank you so much. Today, I feel like I'm back to my home. I always considered Merida as my home, second home. So I'm delighted to talk to the individuals, from my home, okay? And let me also acknowledge Wadi Faculty of Medicine. I've had a long relationship with them. I would like also to thank Dr. Garcia, Dr. Gonzalez, Dr. Castro, and also Marco. Marco is always nice. He puts me in touch with all those IT platforms. So I thank everybody. But also I should thank the audience today, because they are the main recipients of the information I'm trying to relay today. So the outline of my webinar is, first, I would like to give you a quiz, if you agree on that. And then I'll talk, actually, there are only two slides on the background of infection, because we need to understand the background before we go to some challenges, including, how we manage the disease, but also the vaccine issues and variant issues. The last two topics, the vaccines and variants, as you probably know, they are hot global health issues. So I'll be spending more time, on them. So to begin with, okay, this is your quiz. Your patient is 50 years old woman, and she ends up in emergency department. And she has 3 days history of back pain, and she has severe headache. However, 10 days before, she received AstraZeneca virus, okay? That's CHAdOX1, is AstraZeneca virus, okay? So here you see an MRI of uterine, you can see accumulation of clot, okay, thrombin, formation, and this one as well here, and this one as well here. But also it seems, she may have some, intrauterine, clot formation as evidence. Some of them are probably vessels, but these are the lateral vessels. As you know, uterine is highly vascularized, especially laterally, okay? And then they performed a CT scan of the respiratory system, the lungs, okay? And sure enough, it shows right here, okay? It's segmented actually, and perhaps sub-segmented, embolus formation, okay? So the question is, I have for you, before we investigate these, what would be your immediate diagnosis? Based on history, based on two imaging, uterine, and also the lungs. What would be your immediate diagnosis? I cannot see you, by the way. I have only one webcam, but I'm sure, Dr. Garcia or Dr. Gonzalez, some of them can relate to me what was, your impression and what's your diagnosis. Any idea? Any comment? Okay. Who is moderating today? Is it Dr. Gonzalez or Dr. Garcia or Dr. Castro?
So the indication would be for them to write it on the chat. Pregunta el doctor si alguien tiene alguna opinión, si la pudiera comentar en el chat para que se la hagamos llegar, por favor, acerca del diagnóstico probable que se encuentra en este caso clínico que él acaba de describir. So I'm waiting. I'm just gave the indication for them to write it on the chat, but then
yes, because I cannot see the chat. I cannot see the chat.
I can see it. So as soon as I have one or two recommendations, I will give it to you. But then venous thromboembolism would be an option there.
Excellent. Very good. Very good. That's VITT. Okay. This is VITT. That is true. That's venous, and most, scientifically, it is called vaccine-induced, but you said venous, but keep in mind, it's an immune-mediated reaction, okay? It is venous, but immune-mediated, and thromboembolism or thrombocytopenia. So in short, the diagnosis is VITT. VITT. Okay? Vaccine-induced immune thrombocytopenia. So that is our immediate diagnosis. Okay? But let us also work on these questions. The case gave you 4 options. In this case, do you investigate? Do you run a CDC and platelets? Okay. Do you investigate fibrinogen? Do you investigate D-dimer? Or simply you order a venography, or you say, wait a minute, I don't have a PCR. Okay, I need to know whether this patient has reinfection. All right. Or lastly, would you perform a thromboblastography, okay, to evaluate how stable is the clot and how stable are fibrinolysis? So which are 4 options you think would be the best option for this patient?
Bueno, entonces aquí él está preguntando cuáles son de estas opciones, cuál consideran que es la mejor para el paciente. Si alguien, por favor, quisiera contestar en el chat para hacerle llegar la respuesta al doctor. We have no answer as yet on the chat.
Okay, I'll give them a few more seconds.
Yeah, otherwise, an opinion there. Okay, no answer as of yet. Then what I would guess is that we have to study, we have to do a CBC, the count, the fibrinogen, and the dimer concentration.
The answer is correct. The answer is correct. You need to know what is the platelet count, okay? Since there's evidence of coagulopathy here, you need to know what's the status of your fibrinogen, okay? And then as far as D-dimer is concerned, okay, that not also tells you the extent of coagulopathy, but also tells you what is the extent of inflammatory reaction, okay? So the correct answer is first one, investigate blood count, including platelet, investigate fibrinogen, investigate D-dimer, and then that would be your clinical diagnosis. Okay, excuse me. That would be your clinical diagnosis. Okay, so I think this case is really a basis for my presentation today. So let us go to background. Last time I had a COVID webinar with Wadi School of Medicine, okay, I presented this slide, okay. The issue of either the viral illness or vaccine or variants is really that spike protein, okay? This is so far we know up to date, okay? There might be other hidden mechanisms. We don't know. It's a very complex viral disease, okay? But it is also important to know what precisely that spike protein is, okay? First of all, it's not a monomer. It's made of three proteins that stick with each other. We call it trimer, okay? Now, from my 45 years in clinical medicine and being studied viruses and under even Jean-Luc Montagnier when I was in Paris, working in Pasteur Institute in Paris, I'm well familiar with viruses. You know, I don't know if any virus has a spike protein in the shape of a trimer. We have monomeric spike, you know, or dimeric spike. For example, an example of dimeric spike is HIV protein, has glycoprotein 120, which is co-anchored with GP41. So this is a kind of surprising to me that three proteins are attached to each other through sugar. All this yellow, okay, colors there. You see, these are the sugar provides stability. But also keep in mind, if we just enlarge this portion of this spike receptor, okay, and it has two subunits, okay, the S1 subunits, which is involved directly in the binding to the cell receptor, and the S2 subunit, which is involved in fusion. Another word, you know, this one is not enough. Contribution of S2 is involved. In S2 segment, there are several enzymes involved in the fusion of the virus into cell, and one of those enzymes is called TMPRSS enzyme. Okay, so the second background is clinical. The first one was basic science. This is clinical. The question is, all right, the virus has a receptor, which is spike protein, and it contacts the cells. The cells, they have ACE2 protein receptors. ACE2 actually is a homologue of ACE1, okay? So once the virus internalizes into type 2 pneumocytes, because type 2 pneumocytes in the lungs, they have plenty of ACE2 receptor expression, okay? Then what happens upon replication cycle? The virus is capable of damaging endothelial cells, okay? To a point, it causes gaps, okay? Gaps in the endothelial cells. That's where the danger is, because once there are these gaps, the cellular constituents can leak out or in somewhere else. So as a result of that, there is the whole cascade of inflammatory reactions, and these cascades, goes on, including contribution of platelets, okay? And it brings to us a situation, we call it hyperinflammatory state, or you can call it cytokine release syndrome, or simply cytokine storm. But my own terminology is called cytokine madness. I call it cytokine madness, okay? So there are multiple mechanisms, okay, biochemical, pathological, and also hematological going on, all right, which then patients goes into a critical stage, before critical stage, the severe stage, and you have a situation, we call it ARDS, okay? And in this stage, the patient is more susceptible to organ damage, okay? But also DIC, disseminated intravascular coagulopathy. So the question is, what are the challenges of management of COVID disease? And this is very valid for Mexico, okay? Mexico is not doing well. I follow Mexico almost every day, but also Mexico is highly concerned country for the World Health Organization. I've been talking to my colleagues in World Health Organization about Mexico, okay? Because I care about Mexico. I used to come Mexico every year, many times a year, okay? So as I said, it's considered as my second home, especially Merida. I know Dr. Quijano, other doctors, many doctors there, and Wadi Faculty of Medicine. I feel like that's my home, actually. So that's the reason I follow, Mexico. The problem with Mexico is lack of decisions, okay? Mexico has excellent doctors. Mexico has excellent researchers, scientists. But the issue with Mexico, with the lack of vaccination, with the variants that are becoming more and more, common in Mexico, is lack of a robust, okay, political decision. That's what it is, okay? The policymakers, they are making decisions that are inconsistent, not only with the principles of science, but also with the principles of clinical practice. So with this background, okay, at the beginning of viral replication, let's say day 0 to 1 week, at the most 10 days, that is called viral phase, okay? This is the phase you have a high levels of virus detectable by PCR, another word, high viral load. Okay. Now, in the United States, for this viral phase, now we solve the problem. And Mexico can solve the problem. It's not difficult, okay? Yes, if you ask me a year ago how we used to treat our patients, very bad, poor outcome. But since 6 months ago, we found that at least we can take care of this cycle, the viral cycle, by using monoclonal antibodies. Right now, there are 2 of them, the Eli Lilly monoclonal antibodies, and keep in mind, they are therapeutic monoclonal antibodies, and the other one is Regeneron, which is a cocktail. As a matter of fact, in the United States, the Regeneron cocktail works very good on Delta variant, okay? For non-Delta variants, both Eli Lilly and Regeneron cocktail works very well, okay? You remember that Donald Trump was infected with the virus. Well, guess what? He received that Regeneron monoclonal cocktail, and he survived, okay? So the question is, would Mexico be, able financially, or even through World Health Organization or through World Bank, to secure purchase or procurement of these monoclonal antibodies and bring them and make them available to all people, okay? Yes, these monoclonal antibodies are available in Mexico City, okay? But they are thousands of dollars. That's only for rich people, okay? So that does not address the health equity. That's the issue, okay? I'm aware that monoclonal antibodies are available in Mexico City, but it's only for rich people, okay? The health should be for everybody, okay? So aside from monoclonal antibodies, yes, there is one nucleoside analog. I'm sure you heard the name remdesivir, okay? Some hospitals, they use it in the United States, okay? And some they don't. We have 2 large clinical trials of remdesivir. One was performed in Emory Medical Center in Atlanta, Georgia. It's a very good institution. And they had about 9,000 participants in the randomized clinical trials. And the conclusion was it shortens the hospital stay by 3 days. Okay. Well, when you say it shortens the hospital stay by 3 days, it's a kind of non-scientific expression statement. Okay. What do you really mean? Because hospital stay could be, non-ICU, ICU, okay? Then, non-invasive oxygenation, then invasive oxygenation. So you can see there are several confounders it contributes. So that study did not really conclusively address the efficacy. But a couple of weeks ago, there was a large clinical trials of remdesivir. Again, about 85,000 participants. Was a very good, clinical trial, and they concluded from their clinical trials that remdesivir, works in the middle of this viral phase, okay, right here. In other words, it doesn't work when the viral replication is very high, okay? And why it works here, that is a mystery right here. Because if it is a nucleoside analog, it should really target the peak, okay? But it doesn't. So this means, and this is my own hypothesis based on many years that I have experience of viruses and nucleoside analogs, okay? I worked a lot on HIV. This means maybe there are some clinical pharmacokinetic issues, and maybe those issues with respect to remdesivir. Plus, keep in mind, remdesivir is an IV. It is not an oral tablet. So we need to find a drug which would be oral tablet. In other words, when you go to clinical center, health center, let us say Hospital Oharan in Merida, okay? So the doctor will give you pills instead of IV, because IV you have to be hospitalized. So we still need to work in this area of viral, replication, and even though monoclonal antibodies, they're all IV, okay? In other words, monoclonal antibodies cannot be used in a setting of a primary care, although some of our primary care offices in the state of Michigan, they gained approval of the state, and they can infuse monoclonal antibodies under certain provisions, especially has to be under supervision of ID physician, and they can do it in office. All right. Then the next phase is inflammatory phase, okay? Now, this is actually not scientific, all right? Even here, there is inflammatory phase. But once you go further, especially third week and fourth week, these are actually hyper-inflammatory phase. It's not inflammatory phase, hyper-inflammatory phase. That is the reason, okay, then at this point here, the patient develops cytokine storm and eventually ARDS and DIC. Now, as far as management of this phase is concerned, right now we have corticosteroids, okay, intravenous steroids, let us say dexamethasone, 6 milligrams per day. That's the only medication, but there are countries they can afford a second medication, which is called tocilizumab. I'm sure you heard about it, tocilizumab. Tocilizumab is an interleukin-6 antagonist. As a matter of fact, many studies have shown, especially Italian and also the Spanish studies, they've shown that a combination of tocilizumab and dexamethasone can be very effective in the late phase, that is the critical phase of COVID-19. However, I'm sure many of you heard about black fungus, which was first reported from India. India, just like Mexico, has a high prevalence of type 2 diabetes, unfortunately. So many of them are uncontrolled. They're not using medications, or some of them are using medications, but they turn into a very chronic condition. So what happened, they already have existing immunosuppressive condition. So what happened when Indian physicians, they were injecting high-dose dexamethasone to these patients on this period right here, they came up with black fungus, okay? Black fungus is mucormycosis. As a matter of fact, I have a clinical collaboration with India, okay? Without COVID, before COVID, let us say a typical public health hospital, in India would see maybe 1 or 2 cases, per week of mucormycosis. But now all major hospitals, public hospitals, they're saying 5 to 6 cases of mucormycosis. These are basically due to COVID, that they received, high dose steroid. So what will happen, actually the fungus itself is not black at all, okay, if you look at it under microscope. But because it causes necrotic tissue that are black in color in the skin, therefore they call it black fungus. And some of you sitting here may have infectious disease background. You know what I'm talking. Black fungus is not easy to treat. The only medication that we have is amphotericin B. Amphotericin B is not only expensive, but in many countries is not available. This is what India is experiencing right now, because they're either in the shortage of amphotericin B to treat this black fungus, or there is in some major cities, but it's very expensive, and certainly government cannot afford to pay for amphotericin B. So you can see still we have challenges when it comes to COVID disease. Now let's turn into vaccines. Okay, well, there are several vaccine platforms. We have 4 major platforms. One is very old traditional technique, okay, and usually either they're attenuated or inactivated, okay. It's basically called whole virus vaccine, okay. But there's also a new technology, okay, recombinant technology. We've been using that technology for Ebola vaccine, okay. It's called viral vector vaccine, okay. These are like Chinese vaccines or Russian vaccines or AstraZeneca, okay. And these viral vaccine could be Johnson & Johnson, produced in the United States. The third option is nucleic acid. That is the mRNA vaccine, okay. Now, keep in mind, mRNA technology is not new, okay. It's been almost 10 years we have it, especially in the domain of oncology. We have had a couple of, mRNA vaccines, but overall the clinical outcome was not, that great. But it just happened the Pfizer company, at that time BioNTech, a subsidiary of Pfizer, and here Moderna in the United States, they just looked at back in the context of COVID, and certainly, it works, but there are some issues with it that I'll be addressing it later. Then the newest technology is called protein-based vaccine, okay, such as Novavax. This is a Maryland-based biotechnology, okay. So they produce spike protein using insects, okay, insects. So the insects keeps continuously producing spike protein, then they isolate spike protein, okay, and through their technology, separation, filtration, so then they come up, with purified protein, which is called protein-based vaccine, and it uses M matrix, as adjuvant. It's a new technology, but that technology is already for shingles vaccine and hepatitis vaccines. So you can see even this one really is not new technology. We've been using it for shingles and hepatitis B, but now we're bringing it for COVID vaccine. Well, this is fundamental slide. I want you to pay considerable attention because that's the basic immunology we need to understand. So this describes the mechanism action of mRNA vaccine, okay? And this is lipid nanoparticle, okay? LNP, lipid nanoparticle, all right? So we have 2 mRNA vaccines, Pfizer and Moderna. Pfizer, a European effort, Moderna, a US effort, okay? The Pfizer one has 4 layers of nanoparticles. The Moderna one has 7 layers of nanoparticles. Okay, so there's difference between layers, 4 versus 7. But we should all be cognizant of the fact we always talk about B cell, we always talk about helper T cell, cytotoxic T cells, but we don't talk about antigen presenting cells. My good amigos, my good friends, okay, this vaccine must first target antigen presenting cells, okay, before we understand the role of B cells, dendritic cells, and T cells. This is where the literature needs to focus more. We don't have much paper on that, only few. So what happens? These nanoparticles that contain mRNA, they enter antigen-presenting cells, okay? mRNA is released, and then, translated into spike proteins, and these spike proteins are presented, okay, to cytotoxic T cells, helper T cells, and B cells. And keep in mind, okay, in vaccine research, we also need to fully define what is the role of helper T cells with respect to stimulating not only cytotoxic T cells, but also B cell, because if this signal is blocked or this signal is blocked, you simply have spike proteins that are presented to different T cells, but they're just sitting there. They're not doing anything, okay? So in other words, the signal molecules are very important. That signal transduction is very important to make it a uniform response to vaccine. So if any of these pathways are defective, we're not going to have an optimal vaccine response. So what will happen? Finally, the B cells, they produce neutralizing antibody. And keep in mind, I said neutralizing antibody. There are some situations we may have non-neutralizing antibody, which is referred as ADE, antibody dependent enhancement. Well, guess what? If there are antibody dependent enhancement, which they are not non-neutralizing, okay, they facilitate more the entry of the virus into host cells. Then eventually we have memory T cells, memory B cells, and they will hopefully be long-lived. As far as we know, the memory T cells following, vaccination and even infection for T cells, it's quite robust. But if you ask me, is it gonna be for 50 years? I don't know, okay? Our current knowledge allows us to conclude it is long-lived, but we cannot assign how many years. So that is another area of the gap. Okay, so what is the purpose of all these vaccines? Okay, I just listed here the major ones: Pfizer, Moderna, Johnson & Johnson, 2 Chinese vaccine, okay, Sinopharm and Sinovac, the Sputnik 5, okay, and then AstraZeneca. Well, the final assessment of all of these vaccines individually, okay, should be to prevent not only symptomatic illness, but more importantly, this is where Mexico sitting has an issue, and also Mexico in general, to confirm laboratory confirmed infection. Okay. Unfortunately, this puzzle is missing, in Mexico, not only Mexico, and I don't want to attack Mexico. That's not good at all. Mexico has problem right now. Colombia is another one. Okay, Brazil has had so much problems. In the Middle East, Iran is devastated. Okay, devastated. India is another one. So it's not only Mexico. Okay, don't feel so bad about Mexico. Mexico needs a national, independent program of COVID care and vaccine. Okay, the government, the big government in Distrito Federal should appoint an independent, non-biased committee to oversee not only the care aspect, the public health aspect, but also vaccine aspect. That's what Mexico needs, okay? Because Mexico needs to come out of that closet of policymaking, okay? Policymakers, honestly, they do not know well the biology of the virus, the pathophysiology of the virus, and the clinical aspect of the virus, okay? So we need to bring in the doctors, the scientists, researchers, epidemiologists, nurses, in order to bring about a comprehensive solution to the issue of COVID in Mexico. So the question is, these individual vaccines, we call it different platform. Pfizer is a platform. Moderna is a platform, okay? They have a range of efficacy, okay? These are the range of efficacy. And keep in mind, in the last COVID webinar I had with you, with Wadi, I did mention that there is a distinction between efficacy and effectiveness. What you see as efficacy, that's where pharmaceutical companies, they say, let's say Pfizer is 95%. But we know by now, when this Pfizer went into population, millions of population, that efficacy really is not 94% or 95%. Now, the true efficacy, which is called effectiveness, okay, which we call it phase 4. Phase 4 is when it is approved after, FDA and goes into population. Now we know it's around 90%, okay? By the same token, Moderna, it's about 88%, okay? Sputnik is around, 80%, because that's effectiveness, and so on, so forth. So that is the reason you should not really compare efficacy of vaccines, different vaccines. It's simply not scientific. And I will explain to you why is it not scientific, even though they are different platforms. In the next slide, okay, so you can see, for example, AstraZeneca, the type of vaccine, the vector, okay, then they started, the clinical trials, they published it, okay. But what is important to identify is to identify the confounders. So what are the confounders when it comes to location? Let's say AstraZeneca, they only did clinical trials, what I say, it's a major, okay? They had some minor, but let's talk major clinical trial. It was only 2 countries, UK and Brazil, that's it, okay? But look at Pfizer, okay? It conducted, clinical trials in the United States, Argentina, Brazil, and South Africa. But again, don't be misled by list of countries, okay? Maybe this one is valid, but 6%, that's not statistically significant. This is how they mislead you, pharmaceutical companies. You need to pay attention what are not only the confounders, but the real data. South Africa, 2%, statistically insignificant. So we can conclude, really, the Pfizer clinical trials was in principle in USA, and a little bit in Argentina. That's it. These 2 should be excluded because look at the statistics. All right, Moderna, they only did clinical trials in the United States. Okay, and let's go to Sputnik, Russia. They were only done in Russian population. That's it. Okay, but look at Johnson & Johnson. That's American. 1 dose uses, viral vector. Look at it. They did, in USA, in Brazil, South Africa, and Colombia. Yeah, you can exclude Mexico, 1% is not going to make difference. You can exclude Chile, 2% is not going to make difference. Peru, 4%, not acceptable, okay? Argentina, 7%, not statistically significant, but you can at least say it in USA, Brazil, South Africa, and Colombia, it had a valid, statistics, excluding Argentina, Peru, Chile, Mexico, okay? So that's the reason they should not be compared, because a good vaccine is a vaccine that goes through not only different populations, but equal numbers of populations are tested, okay? Then we have, let us say, Chinese vaccines, and primarily they were interested in clinical trials in United Arab Emirates, and 20% in Bahrain. That's the initial. So what do we conclude? There are two conclusions. When it comes to comparison, there are two key factors: time and place, okay? Place, we already gone through it, but look at the time. At the time where AstraZeneca was developing, we didn't have much variant, okay? We didn't have much variant. But at the time where Johnson & Johnson was conducting clinical trials, okay, yes, we have P1 variant in Brazil, okay? We have the South African variant, B1135 variant. So what it is, comparing AstraZeneca to Johnson & Johnson, okay, which are both adenovirus, the data for Johnson & Johnson is scientifically more valid than AstraZeneca, where there was only probably wild type. But here, not only there was a wild type circulating, but at least there were, two variants circulating, Brazilian variant and South African variant. Therefore, my message to you is, do not compare different vaccine platforms. You need to look where they were conducted, the clinical trials, and what was the outcome, what was the statistics, and also, most importantly, you need to look at the time where they were conducting clinical trials. Was there any variant or not? So that's the take-home message. Well, look at this one. Johnson & Johnson, okay? Janssen is a European, okay? Johnson & Johnson is the same, okay? When they conducted their clinical trials, it was 72% USA, okay? And that's acceptable. And in Latin America, in general, turned out to be 66%. Again, it's acceptable, but probably borderline. But Johnson & Johnson is not going to be effective in South African variant because it's close to 50%, okay? So I wouldn't recommend Johnson & Johnson, which is even American-made, in South African variants. Let us say if South African variant is the dominant variant in USA, we can no longer use Johnson & Johnson. We have to go back to mRNA vaccine because it simply is not going to work. Let's take another example. Novavax, which is protein-based technology, okay? When they tested against wild type, that's the original Wuhan, okay, strain, excellent, 95%. Guess what? When they tested against B117, that's UK mutant, the efficacy declined. When they tested against South African variant, the efficacy significantly declined. So Novavax cannot be used when it comes to South African variant. But with respect to UK variant, which is B117, or wild type, good luck on wild type. The wild type is diminishing, okay? There is no original Wuhan wild type, okay? Virus is continuously mutating, okay? So I think we need to understand, exactly, the different aspects, okay? Different aspects, and then make, conclusion. So this is a recent study. About 3 weeks ago, they published it. in Lancet. There were 3 large clinical trials, and studies, UK, Qatar, and also phase 3 in several countries. And there were South African strains tested against the Delta, which is Indian strain, the UK, which is B17, okay, and also the original strains. That is the original variant, okay, detected in all over the world, but also in the United States, okay. This is no longer the dominant one at all, okay. The dominant ones are actually the Delta variant, but also South African variants and Brazilian variant, although we have different kinds. I'll talk about it, So you can see there is a decreased efficacy, okay? And I'm afraid by next year, let us say this is next year here, this curve is gonna shut down here. What it means, we probably by next year or year after need to revise our vaccines. We need to come up with new vaccines to go after some of those nasty variants. I'll be talking about it, okay? So this study was an excellent study. It is giving us an insight that as time goes on, as the virus is mutating very fast, we may have problem, not only clinically, but also epidemiologically. But when you put these two together, the conclusion is, well, we may need new generations of vaccines. So this commentary was published in Nature. It's a very prestigious journal. Okay, it was a commentary. So they put 5 key questions, okay, with respect to vaccines and their side effects, okay. Of course, we know one of the major side effects is formation of thrombus or blood clot, okay. And basically, when it comes to AstraZeneca and also Johnson & Johnson, there were reports of HET, that's heparin-induced thrombocytopenia, okay? But again, we need to go back, if you were attentive on the pathophysiological slide I showed today earlier, the clinical slide, the spike protein really damages endothelial cells. So what happens as a result of that interaction, it provokes, platelets, okay? It activates platelets. So as a result of platelet activation, platelets, they release PF4, platelet factor 4, which is very devastating, okay? Especially there is no regulatory genes to stop. Once platelets, they start releasing PF4, it just goes forever, and that could be, the causes, immune, thrombocytopenia. That was the case we discussed, clinical case that you solved it, The issue of, blood clotting, it's more with vector viruses, okay? So far, we don't have much reported based on RNA, viruses, okay? And then, as far as the blood is concerned, statistically, we know that we have 86 cases reported in Europe, but these 80 cases, they were 25 million, okay? So if you do a little bit of simple math, it's even less than 0.0003%. But it's important because we need to know why is it these 86 cases, they developed, coagulopathies or HET, heparin-induced thrombocytopenia, okay? So we need to understand it. If we don't understand these cases, we will never be able to solve the issues of the next generations of the vaccine. The third question was the group. Well, we know that the AstraZeneca or even Johnson & Johnson, for example, Johnson & Johnson, the report from state of Georgia, there were 7 women, usually between age 17 and 39. They developed, coagulopathies, But again, you need to look at the age group, and the being female, is it really, it's because of their menstrual cycle? Were they in their menstrual cycle? Did it happen after menstrual cycle? Or was it just they were ready to enter menstrual cycle? Or did they have any hormonal irregularities that we don't know? But this article also concluded, because in Europe, they were giving AstraZeneca, especially in Sweden, and the priority were, healthcare workers, and majority of healthcare workers are female. That's why it's reported more in female. Well, that's a general statement, but we need, again, to breaking it down to find out why is it this female, they had bad reaction to vaccine that resulted, either in coagulopathies or head. For example, I have actually a general surgeon who called me, a female general surgeon who called me from Iran. And she told me after she received AstraZeneca, first dose, 2 weeks after first dose, she felt that her right eye doesn't have vision, is losing vision. And she went to ophthalmologist, and ophthalmologist detected a small clot in one of the arteries, of the eye. So, and she wanted to know why is it, happen. I gave her some of these scientific, reasons and clinical reasons why is it, happen. But it just happened. I asked her that, are you using oral contraceptives? And the answer was yes. I told her, maybe this could be a cause. So these are some of these issues needs further investigation before we make any conclusive, statement as to what is it right or what is it not right. Ultimately, these vaccines, with the side effects, reported, they may also have a public health impact. In other words, in general, population may not be interested in having vaccines because they always look at the media and say, oh man, there's this side effects going on, and I'm not going to have vaccine. As a matter of fact, in the United States, statistically speaking, about 18% of population, general population, they are reluctant to receive vaccine. And when you break it down, even the doctors in the United States, 3 to 4% of doctors in the United States, they object to receiving vaccines, let alone we come across with couple reports of vaccines affecting, the ovaries, and the reproductive system. And the first case I gave you, the case of that 50 years old woman, that's a clear example, vaccines may affect reproductive system. So we have a long way to go. Now, another aspect of vaccine adverse effect is myocarditis. I'm sure you heard about it. It's been reported in many journals, okay? So the clinical aspect, the clinical symptom would be chest pain, pressure, occasionally shortness of breath, fatigue, swelling of hands and feet, and part may palpitate, and lightheadedness, and extreme form, severe form, you may have loss of consciousness. But I simply focus on this one, okay? Very easy. Those are cardiologists and attending my seminar today. They know what I'm talking, the endocarditis, myocarditis, pericarditis, eventually they're all the same, and the outcome. It's very easy if the patient is laying down, okay, and gets more pressure, okay? Whereas if you ask patient to sit, that pressure is less, and even the chest pain is less, that is really a typical sign of, myocarditis or endocarditis, or even, pericarditis, usually depends on the severity of the inflammatory reactions going on. By the way, this could be most likely an immune reaction due to spike protein, okay? Whether, through disease or vaccine, this spike protein is very complex, honestly, very complex. And it led me to believe, is it really a laboratory-made receptor or not? Okay, I always have that question, in my mind. Irrespective of the politics, let's focus on clinical aspect. The usual treatment usually there are, IV steroids to tone down, inflammation. But in severe cases, again, those are cardiologists. They know that what I'm talking. Sometimes it needs, IVIG, and to tone down that immune, reaction, But from my perspective, we have a couple cases in Detroit area. They consulted with me. Usually, it's not very severe, and by appropriate treatment, it should, go away, So this is an MRI, and you can see, there are a lot of damage to the muscles of the heart, okay, and at this case, when you have a patient with myocarditis, either with disease or vaccine, it's always good to order troponin, okay, because troponin is a cardiac enzyme. As a result of damage, it's released into blood circulation. The higher level means, the higher, intensity of, myocarditis or, endocarditis, and so on and so forth. So what happens, as we get, mRNA vaccines, and goes through deltoid muscle, and immediately gets in contact with the nearest lymphatic node, okay? And bingo, from here, it gets, in touch with blood circulation, and it is already, hypothesized that those spike proteins may be immunogenic, but also may have site, okay? Site to induce, coagulopathies and also, vascular damage. And this is an outstanding study, which was published in Circulation Research. It's a prestigious journal by American Heart Association. You can see here it's a control cells and a spike protein and versus, a spike protein with SARS-CoV-2. You can see that it causes more damage, okay? It causes more damage to endothelial cell, and these are the blown up of either control or spike protein, okay? So even, if you remove the virus from spike protein, that spike proteins themselves can have a devastating effect, especially their effect is considered to be mitochondrial fragmentation in the vascular bed and leading into endothelial cell damage and the leakage of the vascular bed, and also the activation of platelets, and following the production of platelet factor 4, which then causes immune thrombotic effect. And this is another excellent study done by the Washington University School of Medicine, one of the excellent and most advanced research center we have in the United States. They also demonstrated myocarditis with COVID-19. You can see here the heart muscles are deprived of oxygen here because of inflammation going on. That's a typical of myocarditis. You can pick it up through echo, but occasionally in advanced cases the MRI would be needed. Now, this is a very fascinating slide, okay? Hardly discussed in the scientific community. And that relates to interferon type 1. Interferon type 1, this is an Australian study, by the way. Interferon type 1 is a mixture of interferon beta and multiple subtypes of alpha, Okay. We know that at the beginning of infection, SARS-CoV-2 infection, the production of interferon is antagonized, is suppressed. However, we don't fully understand what is the precise mechanism. Okay? If you remember, Cubans were mass producing interferons and they were shipping plane loads to China at the beginning of pandemic, and the Chinese were using interferons, but with no clinical outcome. As a matter of fact, a couple of Chinese report indicated even it worsened the outcome, because interferon itself can induce inflammatory pathway leading to cytokine storm. Okay, it's been well demonstrated in macrophage as well. Okay, so, and also we know that it's a very interesting slide. I love this slide. In influenza, okay, seasonal flu, our patients, they mass produce interferon type 1 to the point they get, viral pneumonia. You have to treat viral pneumonia. Very bad. Okay. However, in contrast to influenza, the interferon type 1 suppression is suppressed or antagonized in SARS-CoV-2, in COVID disease. 2 respiratory pathogens with 2 different interferon type 1 profiling. Okay, that's number 1 issue. Number 2 issue is that we know that mRNA vaccines indeed produces interferons. Okay, indeed produce interferons. And this is very interesting. Many reports indicates young people and female, they produce more interferon type 1 if they receive mRNA vaccine. Okay? That is the reason could explain some of these myocarditis, endocarditis, others, because interferon can signal inflammatory pathway. Okay? But in some individuals, we don't see hyperinflammatory response. Even a low level of interferon, the low level of interferon, as a matter of fact, is protective. Okay? So you can see here the mystery, the complexity of science, when it comes to, let's say, pharmacokinetic dose, higher dose, a different response versus lower dose, opposite response. Okay? So what it means that, is it possible, for example, after a booster shot of mRNA vaccine, okay, this may go higher and higher? Or is it possible a patient has mRNA vaccine, all right, but is reinfected with a new variant of SARS-CoV-2? Is it possible that that virus can suppress the production of this interferon, which is induced by vaccine? So these are the questions. Honestly, we don't know the answer, but these are good clinical research projects. The other thing is, this is just a week ago, a very nice paper came from Nature, okay, entitled SARS-CoV-2 mRNA vaccines induce persistent human germinal center response. Excellent study. I love this study, okay? So what they did, these are the arm with SARS-CoV-2 infection. They received vaccine, okay? And these, okay, that they had a history. So this is without, the red is without, this is with SARS-CoV-2 infection. So you can see without SARS-CoV-2 infection, they did a little bit less effective than SARS, history of SARS-CoV-2, okay? Very interesting. Although you can see that the IgG remains quite high even after 15 weeks. That's why, many people ask me, I do consulting, clinical consulting with many countries, at least a dozen countries. They always ask me about this. Well, this is the limit of the study, but if you were to hypothetically draw the curve here, it cannot come down sharply down here, okay? It may continue for many, many weeks, okay? So that's the reason, we may not need a booster shot with mRNA vaccine at this point, okay? And actually what they did the S-binding protein and also receptor binding protein, IgG. You can see the data is pretty much similar, okay, with respect to IgG and RBD, IgG. This was an excellent study. And so far we know that Pfizer and Moderna, okay, they can sustain long-lived plasma cells. Based on this study, memory B cells can be long, but we don't know, either a year, 2 years, 5 years, or lifelong. It's too early, okay? With respect to neutralizing antibody, okay, we know that these are specific neutralizing antibody, but again, you heard me at the beginning of this seminar that there might be individuals, they may not develop neutralizing antibody. They may develop non-neutralizing antibody, the so-called ADE. So we need to investigate. As a matter of fact, ADE in dengue is very common, not only in infection, the dengue infection itself, but also, in dengue vaccination. So you can see we are getting, a little bit better on vaccines. Hopefully, new generations of vaccines can cover some of these concerns we have. With respect to variants, well, currently the dominant variants globally is B117 UK, B1352 South Africa, P1 Brazil, and then the Delta variant, which is B16172 India. That's the latest one. But really, we shouldn't focus on these, whether it's P1 or B1 or country of origin, okay? What is important, let me emphasize, to look into if the genome has either deletion or addition or substitution. That is very important, okay? For example, with B117, okay, we have deletions and we have substitutions. With South Africa, which is more devastating because that's a very bad combination. Let me emphasize that E484K and N501Y, this is a very devastating combination, okay? That's where the efficacy of monoclonal antibody diminishes. That's where the efficacy of vaccine diminishes. That's where, the disease is more contagious. And that's where you receive more hospitalization, okay? They have the Brazilian, it's the same, okay? It's more transmissible. But then you can see that the Indian one, the Delta, required additional. That's the more devastating one, okay? Yes, there's a loss of N05, okay? Eventually, I won't be surprised in next month, you can add here N501Y, but this one is the more dangerous one. That's why some of these vaccines, they don't have efficacy, the mRNA vaccines have efficacy of about 80 to 85%. The efficacy of Johnson & Johnson is questionable. We need more study. Efficacy of AstraZeneca, the Chinese vaccines and Russian vaccines, they are totally out of questions because there's no study about them. So what are the variant of concern? These are the Alpha one, which is the UK, Beta one, which is South Africa, Gamma, which is Brazil, Delta, which is the India one. Now, in addition, we need to understand that we in the United States, we have Epsilon variant, which initially described in California, That's the mutation at 452, 2 amino acid substitution, okay? And we know that this type of mutation, the vaccine efficacy is reduced 3 to 6-fold, okay? But also we know that, if we treat these patients with monoclonal antibodies, the Eli Lilly is not effective, that's a monoclonal, but the other one, Regeneron, which is a cocktail, it's effective. So with respect to Delta variant, 10 days ago, I wrote an opinion. I sent it to actually Dr. Garcia, Dr. Castro, and if any of you interested, I'll be happy to send you. If you have my email, I'll send you. In this commentary I wrote 10 days ago, I did mention that the Delta variant is going to be probably a dominant variant all over the world. Okay. Now, clinically speaking, Delta variant has a little bit different presentation. Let us say the original wild type or other variants. This one is more really a typical flu-like symptom with a lot of runny nose, and 60% of the patients, they complain of sore throat. Okay, sore throat. So it has a little bit different clinical manifestation than others. And it shares actually one amino acid substitution, that is L452R, with epsilon. With respect to Brazilian and the South African, although it shares E484K variant, okay? So that means this mutation is shared by Delta and also with Brazilian and also with South African variant. With the original variant B117 from Kent, UK, it already gone through 2 deletions and 8 substitutions. This is a slide a little bit old. I won't be surprised. There might be more deletions here and substitutions, okay? But what is important to understand, this is more contagious if the substitutions happens at N501Y. That makes it more transmissible, okay? And this is the top view of the receptor. You can see that, the 501 substitutions occurs on the top. Basically, this is the receptor binding domain, okay, which makes it, very difficult to receptor to bind, to the antibody, Therefore, it has a higher rate of, transmission, As a matter of fact, the Delta variant supposed to be 56 times more transmissible compared to the UK variant. Well, this is the diagram of the receptor. And that is the spike protein, and then the ACE2 receptor. You can see if there is a substitution at 501, that makes it very difficult, to receptor to bind. Therefore, the virus keeps, replicating more and more organ damage. This type of mutation is now reported all over the world except North Pole and South Pole. You have it in Europe, Asia, North America, Latin America, Africa, and South Africa. But also, you need to understand that these spiked receptors, they undergo deletion. Well, when we hear deletion, that doesn't mean a good news, actually. Some of these deletions can be very successful, leading to more transmissible and more contagious virus, okay? So basically, these deletions, they change, they bring conformational change to the structure of the protein and has always potential to make it more transmissible. The deletion was first detected in Denmark, in animals. As you know, minks, they carry the virus a lot, and that's the reason the Danish government, started killing many thousands of animals in Denmark, which brought, protestations of animal rights groups, so on and so forth, So the other aspect is the South African versus Brazilian. Yes, they share a mutation which is called E484K, which makes it very dangerous, very transmissible, and very contagious, and therefore more hospitalization. So the question is, what are the outcomes of these new variants? Okay, we can classify them in 3 outcomes. 1 would be more transmissible, though they resist to antibodies, or it could be a combination of 1 or 2. That is very devastating. That means hospitalization and perhaps even ICU at the clinical level. Well, the UK variant, okay, we know that can be actually one of the variants of dominancy, having dominance in the United States, even in Mexico, okay? But you should keep in mind, Delta is coming too. And here I'm showing, the antibody that binds, to receptor, okay? And this is the N-terminal, and then the ACE2 receptors, So these variants, what we do, they circumvent this process. The antibodies, cannot, attach themselves to the virus, okay? The most important aspect of especially the South African and the Brazilian variant is that RBM, okay, receptor binding motif. Because they're highly variable, therefore they're susceptible to mutations. And that is in the S1 portion. As you know, the receptor has S1 segment and S2 segment. S1 is basically attachment and S2 is the fusion. So because these regions, which is part of receptor binding domain at the molecular level, are highly variable, therefore they undergo selective pressure. And that is the most dangerous form of mutations. So there are 3 likely scenarios. 1 is a strong neutralizing antibody, either disease or vaccine. No problem. The patients will survive. Those who got vaccine, they produce the right antibody, could last many years, or sometimes even depend, could be many, many years. But there are 2 dangers. What are the 2 dangers? 1 would be intermediate production of this antibody. In other words, suboptimal, okay? Well, that creates escape constructs, okay? So the virus may escape because it's not adequate, the response. The worst outcome is a combination where you have a high viral replication, that means high viral load, okay, and intermediate level of antibody production, meaning neutralizing antibody production. That's the more devastating form, which, eventually these cases end up in the hospital, okay? AstraZeneca had a very tough year this year because of the vaccine adverse effects, and they had to stop vaccination in Canada, for your information. Now in Canada, for the 2nd dose of AstraZeneca, they're offering mRNA vaccines, either Pfizer or Moderna. Many Canadian doctors and scientists, they contacted me and they wanted to know whether it's okay. I told them, look, you should look at the outcome of any vaccine. I don't care, Chinese, Russian, Mexican, US, European, should be whether, number 1, they can maintain adequate level of neutralizing antibody. That's number 1. Whether they can maintain cytotoxic T cell activity, number 2. And whether the neutralizing antibody produced can resist, okay, new variant, that's number 3. Most importantly, number 4 is whether they can sustain in years. So if these 4 criteria are considered, I personally really don't care whether it's Chinese made or Russian made or American made. So I basically told them it's fine. they can use, mRNA vaccine as the second dose. Johnson & Johnson, well, although the efficacy is lower, but again, I told you, the lower efficacy doesn't mean a bad efficacy, because Johnson & Johnson started late in their clinical trials. They included variants, whereas the original mRNA vaccine primarily was done in an environment where wild type was still dominant, okay? So it's a one dose. It's very practical, especially in Mexico, in remote area. It doesn't need refrigeration, okay? It's only single dose. You don't have to go back in remote control areas, villages, to administer second dose, only one dose, And it's really ideal for young people. And the reason is that young people, they do not develop full-blown disease, okay? So considering all this scientific, data I gave you, it's not a bad vaccine, but there is one major question about Johnson & Johnson, whether it works against Delta strain. I don't know. There is no study, okay? And if it works, at what level it works, again, okay? So, but if you exclude the Delta variant, okay, and also South African variant, remember that slide I showed you, it's not really very close, efficacy to South African, but in an area where there is no South African variant, or Delta variant, it works well, okay? So I'm just coming to the end, I wrote actually an opinion about 3 months ago. I sent it to both Dr. Castro, Dr. Garcia, and I did say in my opinion that this spike protein is very complex, and I felt myself in my hypothesis that spike protein may have certain regions, that these regions, can induce some side effects, can induce endothelial cell damage, but also I concluded these regions may also be responsible for coagulopathies. Until 2 weeks ago, I came across with a publication, okay? Although this is preprint, okay? It needs to go through peer-reviewed, but basically it confirms what I hypothesized 3 months ago, and right now we understand that the spike protein itself has capability to induce, coagulopathy. And again, that is a mystery, and that's why perhaps, we need to look into it. Is this really a real biological receptor or a type of receptor which has been manipulated in the lab? Now, this slide refers into quality control. Okay, this was actually a case report. The patient had diabetes, okay, and they developed, pulmonary, disease. We then got, into COVID variant, okay, and so ended up in hospital, and they were interested, actually, this is actually a British study, they were interested in collecting, the samples from this patient for genomic analysis, okay, and see, what kind of variant is involved with this patient, because the patient had a couple pre-existing histories, and it was a good candidate for a translational study. So what they did a whole genomic sequencing. That's what it should be. I don't know Mexico has it or not, but I contacted CDC. They tell me many Mexican samples end up in CDC lab for analysis. You're going to have to tell me, okay? So at the end, they collected 95.6% of genome for GS, that means, genome sequencing, okay? Now, I was not able to put the whole, patient history here, but 2 weeks before that, they collected a sample which turned out to be 82% of the whole genome, okay? 82% whole genome. That when they did a whole genome sequencing on 82% of genome collection, it did not show listen to this, it did not show any S1 mutations in the region. Interesting. It did not show. But this this group was very strong. Okay, they said, okay, well, let's wait until we collect almost the entire genome. Okay, almost entire genome, which is closely to 96%. Guess what? When they collected this genome, okay, which is closely to 96%, and they did a whole genome sequencing, then they found out there is a mutation. So what's the take-home message? Okay. Reinfection has been confirmed before a handful of cases. Okay, this was a reinfection. Okay, so what is telling you, okay, but reinfection confirmation relies on whole genome sequencing, so the cases will be drastically, determined. So this, what it means that we are doing genome sequencing even in the United States, okay, Mexico, Brazil, India, anywhere else. If it is not close to entire genome, it invalidates our interpretation, okay? It invalidates our interpretation. So that the take-home message for Mexican scientists, especially laboratory investigators, is that you really need to collect the whole genome in order to see what kind of variant is involved. I thought that study was very interesting. So what does it mean? The disease, mutations, vaccine, treatment, early treatment, hospital treatment, ICU treatment. That all leads us to one single concept, and that is the concept of epigenetics. This is a disease I spent time since the beginning. Our Mexican colleagues are aware. I'm the first doctor in Michigan wrote about it, okay? Even it became pandemic. I didn't have name for this virus. I called it Wuhan viral pneumonia. I published it in Journal of Infectious Disease and Tropical Medicine. And I did say in that publication, if this virus indeed uses ACE receptor, that could be devastating for the whole world. And indeed, my prediction was correct. But through all these variabilities we have with respect to disease itself, manifestation itself, lack of appropriate treatment, both in early stage and the later stage, although in early stage we do have effective therapeutic monoclonal antibodies, it leads me to believe that epigenetics is very important. So we need to go back to study epigenetics at the DNA level of individuals. Of course, we cannot study the whole population of the globe, but each country needs to select, to handpick different ethnicities of their own, look at epigenetics, either through the disease or vaccination or therapies, and come up with much better and more scientifically sound answers. So in conclusion, epidemiologically, these variants, they pose global risk, but two mutations are extremely dangerous, highly pathogenic, highly transmissible, highly contagious. One is E484K, the other one is N501Y, or the combination. Especially this one, E484K, it evades immune system, okay? It evades immune system. And keep in mind, when it comes to vaccine efficacy, please do not compare them, because as I explained to you, there are several confounders involved, okay? Especially what ethnicity, what different groups, and whether the variants were involved or not, And but keep in mind, we do need, new generation of vaccines to address, the efficacy with respect to new variants, okay? And vaccines may cause harm, that's true. But when it comes to risk versus benefit, currently our benefit significantly outweighs the risk. But overall, my good friends, there are several challenges that remain to be challenged themselves. Thank you so much. And let's open this for questions and comments.
Well, thank you for your excellent presentation, Dr. Nassiri. I've been asked to give a brief summary in Spanish of your presentation.
Yes.
And then we'll proceed to the questions. Me pidieron hacer una síntesis muy breve, creo que es un reto gigantesco con la cantidad de información que se presentó y sin el apoyo de diapositivas. Hablamos ahorita del impacto clínico y epidemiológico de las mutaciones de SARS-CoV-2. El doctor abrió la presentación hablándonos de ciencia básica y de ciencia clínica, dándonos la relevancia de la proteína spike y de las mutaciones que ocurren en esta proteína y reconociendo que esta proteína en realidad no es un monómero, sino que algo que es atípico en los virus, es que es una proteína de 3 componentes que se llama un trímero. En la clínica resulta que esta proteína spike es la que se une al receptor de ECA2, por esta vía se internaliza a las células y dependiendo de dónde ocurra, principalmente en pulmón, va a generar daño en el endotelio. Aquí dependeremos entonces de la intensidad de la respuesta inmune para conocer la manifestación que se presentará en los pacientes. Si esta respuesta inmune es intensa y ocurre una gran respuesta inflamatoria, habrá un síndrome de liberación de citocinas que generará daño endotelial y ocupación alveolar. De manera tal que aquí vamos a tener no solamente fenómenos inmunes, sino al tener el daño endotelial también habrá activación de la cascada de la coagulación que lleva a manifestaciones hematológicas, ambas conduciendo probablemente y otra vez dependiendo de la intensidad de la respuesta inflamatoria a síndrome de dificultad respiratoria y a coagulación intravascular diseminada, lo cual explica el cuadro. Sabemos que en el contexto de la infección viral tenemos, desde que ocurre la infección, una fase subclínica en donde la infección viral es indetectable y en los primeros 10 a 14 días o 2 primeras semanas ocurrirá una fase de replicación viral en donde a los pocos días será positiva la PCR. En este contexto, el doctor Nassiri mencionó como alternativas de tratamiento terapias de anticuerpos monoclonales, una de las cuales es conocida como cóctel Regeneron, que él mencionó que se le administró, por ejemplo, al expresidente Trump, que están disponibles en México, pero que representan un costo significativo y que podrían ser una alternativa para disminuir la replicación viral, por lo tanto, la manifestación de la enfermedad. Ya cuando el paciente se encuentre, otras alternativas de tratamiento que han sido probadas incluyen al remdesivir, cuya eficacia se demostró, se reporta en un amplio estudio con 85,000 pacientes, con la reducción hasta de 3 días de la hospitalización, sin mencionar si este impacto incluye resolución de los ingresos a terapia intensiva o de la necesidad de administración invasiva de oxígeno, o simplemente estamos hablando de días de hospitalización simplemente sin más datos. Cuando el paciente ya ha pasado las 2 semanas y si es que prosigue a la fase inflamatoria o de cascada inflamatoria con tormenta de citocinas, entonces estará hospitalizado y los únicos tratamientos que han probado eficacia son el tratamiento con esteroides, esencialmente dexametasona a 6 miligramos día, o tocilizumab, que es un antagonista directo de la interleucina 6. ¿Qué es lo que hemos encontrado con esto? Bueno, que dependemos en los tratamientos específicos monoclonales o remdesivir de la cuestión de costo y que hasta el momento esos son tratamientos que se administran vía intravenosa, por lo tanto requieren hospitalización, aunque el paciente se encuentre en buena condición clínica, y que en el tratamiento con esteroides en poblaciones, esto se demostró en la India, pero es esperado también en México, que tienen alta tasa de comorbilidades como la diabetes mellitus, se incrementa el riesgo de infecciones derivadas del uso de esteroides como la mucormicosis, que ha tenido reportes importantes en la India. Una vez que hemos repasado el cuadro clínico, el doctor pasó a hablar de las vacunas, en el entendido de que existen esencialmente 4 plataformas, la de virus atenuados o inactivos, la de recombinación, la de vectores virales, la que está basada en proteínas y la que está basada en ácidos nucleicos, en RNA mensajero. Para poner un ejemplo, hablo luego de la vacuna de Pfizer y Moderna, que está basada en RNA mensajeros. Lo que se hace es que se mete el RNA mensajero en una nanopartícula lipídica, se presenta esta nanopartícula lipídica que porta el RNA mensajero, llega a la célula presentadora de antígeno, se internaliza, entonces se quitan las capas de nanopartículas lipídicas, se ingresa el RNA mensajero, se lee, se sintetiza la proteína spike, se presenta esta proteína spike a los CD4, a los CD8 y a las células B, y es entonces que se genera la memoria y los anticuerpos neutralizantes. Lo que no sabemos con que es uno de los varios mecanismos que van a llevar a la misma consecuencia, es el tiempo de duración de la protección. ¿Qué es lo que estamos buscando con estas vacunas? Cualquiera que sea la plataforma, es la prevención de infección sintomática, pero también la prevención de enfermedad confirmada por laboratorio. Y se han reportado eficacias que rondan hasta el 95% para Pfizer, hasta el 60% para Astra y en todo el espectro. Sin embargo, para interpretar estos valores de eficacia, no solamente debemos de ver los reportes, sino también tenemos que conocer en qué población se hicieron estos estudios, porque diferentes poblaciones tendrán diferentes comportamientos clínicos, y también el momento en el que se hizo el estudio, porque cuando los estudios se hicieron antes, digamos el año pasado, probablemente no existían las diferentes variantes, lo cual nos va a dar cifras falsamente más elevadas de eficacia. Y entonces estudios que se han hecho más recientemente tienen tasas menores de eficacia porque tenemos poblaciones que probablemente ya están presentando variantes diferentes. Y habrá que recordar que como estamos presentando una proteína spike, la inmunidad se genera específicamente contra la proteína que se le presentó al paciente. Y si han habido mutaciones, entonces vamos a tener quizás proteínas diferentes que van a ser más difíciles de reconocer y de neutralizar por el sistema inmune. Por esta misma razón de que las mutaciones van implicando cambios en la proteína que es reconocida por el sistema inmune, se puede prever la necesidad de que las vacunas al cabo del tiempo vayan a requerir actualización en su síntesis para dirigirse contra estas nuevas proteínas o estas variantes proteicas, de manera tal que el sistema inmune pueda reconocer a lo que ahora se vuelva la variante dominante de coronavirus, determinada por las características genómicas de su proteína. Le habló de las variantes alfa, beta, gamma, delta y ahora epsilon que está surgiendo en Estados Unidos, y realmente, como les acabo de decir, estamos haciendo referencia a que cada una de las variantes que se menciona se categoriza así porque ha cambiado de alguna manera la secuencia de los aminoácidos en la proteína spike. Y al haber modificaciones en la proteína, se espera que el sistema inmune sea menos capaz de reconocerla, incluso por haber tenido infección previa o incluso en los pacientes vacunados. La otra implicación es que debemos de recordar que las mutaciones ocurren en función de selección natural. Entonces, cuando un paciente ya ha recibido vacuna, ya ha desarrollado anticuerpos neutralizantes en forma baja o intermedia, o ha recibido un esquema parcial de vacunación, pero se introduce el virus, entonces aquellos que son detectados pues son eliminados, pero si el virus muta o es una variante mutante, puede que el sistema inmune no reconozca en absoluto o no reconozca con la misma eficacia a estas nuevas proteínas que son diferentes a las que le presentaron a los linfocitos B y T, y entonces esta variable se empiece a replicar y genere nuevamente infección. Y esta es la lógica que explica cómo es que se están haciendo nuevas variantes más prevalentes. En este caso, lo que se espera actualmente es que la variante Delta es la que se haga predominante, pero no tenemos información de lo que va a ocurrir en el futuro y solamente podemos poner. Por último, y teorizó el doctor de la irrelevancia de la epigenética. La epigenética sabemos que es la regulación de la expresión del genoma, la cual se modifica en función de la exposición ambiental. Sabemos que la epigenética entonces es relevante en este sentido, que la respuesta que las personas van a tener a la infección es determinada por esta forma en la que se regula la expresión genética, y que quizás esta es una explicación de por qué a diferentes edades la presentación clínica es diferente, puesto que conforme va avanzando la edad se modifica la forma en la que la epigenética regula la respuesta inmune ante las infecciones. Y esto es un campo que él propone como interesante para investigación. Espero que esto haya sido una síntesis satisfactoria de lo que acabamos de escuchar. Pasando a esto, determinando esto, abrimos el espacio a preguntas que esperamos en el chat. I just presented a small synthesis of your presentation. Hopefully it was clear enough for the audience. We do have a question that was written beforehand. The question is this: are socio-demographic conditions being considered as a factor in efficacy of vaccines? rather than just variants. And by socio-demographic conditions, he means income, education, and access to medical care.
Yes, actually, there are few publications on that. One by British group, the second one by Indian groups, and the third one by Australian groups. The so-called socioeconomic brackets, and if I read this correctly, would socioeconomic, disadvantage puts you in inefficacious response to vaccine? Am I correct?
Yes, I think that's the spirit of the question.
Yes.
Would a less developed population be more at risk of having less efficacy of the vaccine?
Yeah. Well, this is a very good debate. As a matter of fact, I attended a similar debate on a meeting of WHO, which is virtual, by the way, and we discussed socioeconomic, brackets of the whole world, not only in the context of vaccine, but also the disease, itself. We do have some preliminary data that the socioeconomic disadvantage, or this doctor says social demographic condition, that's basically socioeconomic disadvantage. They, especially they're female, they are, for some reason we don't understand. We need to look into it more detail in the science of it, that they do not respond well to vaccination. Okay, so is there an immunological, issue? Is there strictly a biological issue? We don't know yet, but that's certainly one of the top hot issues that we're discussing with the WHO. And this is very evident, actually, not only in Brazil, but also in India.
Okay, thank you. There's another question here. Would you recommend waiting until there is an effective vaccine against the new variants, or would the recommendation be that this person gets the Pfizer vaccine, which is the most effective vaccine up to this point?
Excellent question. I received 2 questions. I wouldn't wait, honestly.
Wouldn't wait.
If you have Pfizer, take it. If you have AstraZeneca, take it. Don't worry. As I said, I have scientifically demonstrated the adverse effects, even coagulopathy or thrombocytopenia, the incidence is less than 0.003%. Okay, and some of them are manageable. Myocarditis, honestly, is manageable. Okay, or if it is het immune thrombocytopenia, IVIG, manageable. Okay, so don't worry about those unusual side effects. My recommendation for my Mexican brothers and sisters is get the vaccine, whatever vaccine is available. Okay. However, when it comes to Chinese vaccine, we already know they have lower efficacy for sure. Okay. But there are some countries they're using Chinese vaccine. I consulted for the health ministry of Chile and Turkey, and they were very much interested in getting a Chinese vaccine because it's cheap. That's for sure, But I told them, be careful, because it may work in some population, may not work, And so what happened, I know, for example, in Chile, they had to go buy Russian vaccine because some of those received Chinese vaccine, they were reinfected. Okay. Or in case of Turkey, I have more, contact with them. 30% of their population received Chinese vaccine, full dose. They got reinfected. Okay. So my suggestion is avoid Chinese vaccine as much as you can in Mexico. But if there is any other vaccine, take it and don't worry about side effects. Mexico has an excellent doctors. They can manage side effects.
Then there are 2 more questions, but I'm going to add to this one. And we do know that some vaccines are less effective than others, but then low protection is way better than no protection at all. And since we are unable to choose which vaccine we get, then it may be reasonable to get whatever you have access to and then revaccinate if possible, or whenever the new vaccine or the second vaccine or the actualization.
I agree with you. As a matter of fact, Pfizer is coming with second generation, which covers, all these variants. Okay. And also yesterday I had a communication from Johnson & Johnson. They are revising their vector vaccine to a point, where they're including all these variants in the genome, in that case DNA, because mRNA is different. So rest assured, by next year, 2022, we will have a new generation of vaccines that they can cover, all these emerging variants. But also keep in mind, I don't want to give 100% hope. This virus is always mutating. And as I alerted in my presentation, we should expect, emergence of new variants. But again, our technology is very good, okay? And what I mean, it's not necessarily United States, okay? Every country has their own scientific, strength. But the good point is we are alert. We have a good molecular epidemiological methods and assays. We can detect immediately, like these high-level sequencing, they really helped us. If this was, let us say, 20 years ago, I would say half of the population was devastated, wiped out, okay?
Yes.
So we have advances, and we're monitoring, but as I said, there are many gaps, and there are challenges, needs to be addressed. And my 2 cents to offer for my Mexican brothers and sisters is that don't worry, technology is on our side. Get vaccination, including Chinese if it is available, okay? And then we'll figure out, what would be the new generations of vaccines that would be more effective.
While these new vaccines become available, do you expect that we'll start to see cases of severe disease and maybe deaths in vaccinated population due to the appearance of new variants?
Absolutely. The problem we have is not vaccinated people. There are many unvaccinated people that resist vaccination. Those are the most dangerous population that can spread any kind of virus, okay, any variant. As a matter of fact, those populations that resist vaccination, they may have a combination of several variants, and they can transmit, okay? It's not surprising to see an unvaccinated individual that has a variant of Brazilian variant, South African variant, and Delta variant, all three in the same blood.
On the same patient,
yeah.
Do you expect, when you say that there is that certain variants, certain vaccines are ineffective for certain variants, do you mean that you will have the same clinical scenario, or would you rather say that being vaccinated would in fact reduce the intensity of the clinical picture?
The second one, it will reduce the intensity of clinical picture.
So even if you have the disease, you will not have it full blown. You will not have the cytokine storm, right?
From our studies,
you're less likely to get it.
Yeah, from our studies, especially with the mRNA vaccines, Moderna and Pfizer, we really don't have any reports so far. I mean, in the United States, especially in our hospitals in Michigan, that so-and-so got vaccinated with mRNA vaccine, or even Johnson & Johnson, they ended up in ICU. The incidence is very rare, very rare. As a matter of fact, we did some preliminary data on that, If it happens, the patient has so many other comorbid conditions that allows him to end up at ICU. But if there is no comorbid condition with these vaccines, we never see any patient at ICU at all. They do not require mechanical ventilation at all.
You may get sick, but you will not die.
No, you won't.
Which is a good outcome to be fair.
Well, it is a very good outcome because we prevented mortality.
Yeah,
Okay?
and then
At least by mRNA vaccines.
In your previous presentation, you did tell us about the definition of efficacy, and you can define efficacy by not dying. You can define efficacy as not getting to the ICU, or by not needing to be admitted to the hospital, or by not showing any signs. So it depends on how we define efficacy, and then even those vaccines that are not showing 95% efficacy against disease are still effective on preventing death. That's the take-home message.
There are several confounders. One would be hospitalization, okay? The other one with prevention of disease. The third one is transmission, okay?
Yes.
We achieved the goal of hospitalization and disease prevention. When it comes to transmission, we still have issues, okay?
Yeah.
And that is the reason I always tell whether people are vaccinated or not vaccinated, wear masks.
Yes.
Until next year, we figure out what is going on, okay? This year, we did many progress, no problem, okay? But you saw from my presentation, we still have some gaps. Because we have gaps, that means the transmission is likely in those gaps. So what would be the public health approach? To use face masks, at least for another year.
Yeah, because as you pointed out before, if you are in fact vaccinated, you can still get infected. And if you get infected and you are vaccinated, you are a place in which an additional mutation can occur, right?
You asked a brilliant question. A brilliant question. Let me. Even in vaccinated individuals, okay, when, let's say, hypothetically, they get reinfected, okay, they may have suboptimal infection, okay? That means they are asymptomatic carriers, okay?
Yes. Okay.
That means, I mean, clinical scenario, it doesn't happen oftentimes. So that suboptimal infection, which clinically is asymptomatic carrier, okay, even if they don't use face mask, if they go out, they sneeze, they cough, they can give that virus to individuals who are not vaccinated. You see what I mean?
Yeah.
So what is the take-home message here? Those vaccinated, those unvaccinated, definitely in the hospital, they all should wear masks until next year we figure out a better approach to prevention and the control of this virus.
Okay, thank you. Then there's the last question.
Yeah.
From Dr. Garcia.
Hello,
Gamma and
Dr.
epsilon
Garcia.
variants are predominant in Mexico. Do you know if current treatment that you talked to us about work in these variants the same way?
Yeah, the gamma and epsilon, if you diagnose it early, okay, and I suggest for gamma and epsilon, use monoclonal antibodies, okay? And I don't think it's available in your hospitals. It's very expensive, okay? I know a couple companies in Mexico City, they have it. Injections are more than $10,000, okay? So I am willing to work with Wadi, okay? If we could make a paper, white paper, not publication paper, white paper, and through the agency, federal agency, submitted to World Bank. I know a couple other countries, they do it. So the World Bank give Mexico a loan only to buy the therapeutic monoclonal antibodies from Regeneron and Eli Lilly and make it available to public hospitals. You know, that is doable, honestly. I mean, World Bank is very, very receptive because we are in pandemic. They cannot reject the request of Mexican government, you know. And if you're willing, your governor, okay, I knew the past two governors of Yucatan, I don't know this one, okay, And we can have actually a meeting through Skype. And I'll be very happy to write a white paper, although it will be English, then you have to translate it, in Spanish. And through your governmental agencies, you can submit it to the World Bank. I know that you can get about $100 million, which is not bad. That can cover some of your hospitals. By the way, we can always negotiate with Eli Lilly and Regeneron to give you a discount, because of the NAFTA. See, you have to bring in, some of these, conventions you have. NAFTA makes it easier for Mexico to receive monoclonal antibodies for this infection. Yeah.
Okay. Well, thank you for your excellent presentation and for all your time and your availability to answer all of our questions. I will give the word to Dr. Garcia just to close the seminar.
I see Dr. Garcia. Hello, Dr. Garcia.
Thank you so much, Dr. Nassiri. It's an excellent presentation. I think so. I hope one day we're gonna produce my vaccine. What in Michigan State, right? So,
well, in Michigan, we produce Pfizer in Grand Rapids, by the way.
Okay, thank you so much. Gracias, Dr. Pablo. Gracias, Juan. Thank you. And you're welcome. We need to stay here to Merida. I hope next year we're gonna go to visit your second house. This is your home.
Yes, it is my home. Merida is my home, really. Yeah.
Thank you so much.
No problem. And looking forward to news, maybe webinar, maybe in 3 months or 4 months when we have more information. I will alert Dr. Garcia. Okay.
Thank you.
And hello, Dr. Cetina.
Muchas gracias, profesor Reza Nassiri, por compartirnos pues todos sus conocimientos, experiencias y opiniones sobre este tema tan relevante el día de hoy, ¿sí? De igual forma, agradecemos a todo el personal involucrado en la planeación y ejecución del evento del día de hoy. Los invitamos a seguirnos en todas nuestras redes sociales, Facebook, Instagram y YouTube, donde podrán volver a ver esta transmisión del webinar, así como mantenerse informados sobre los eventos que tenemos próximamente. Muchísimas gracias a todos y que tengan un buen día.